Alrighty, it is that time, so I think we will commence just so that you know you're not in the wrong room. This is Symposium 30, GPCRs in Addiction. My name's Andrew Lawrence from the Florey, which is at the University of Melbourne in Australia, and my co-chair is Chris Langmead, who's just up the road from me in Parkville at Monash Institute of Pharmaceutical Sciences. So we're co-chairing this session. I think it's going to be an exciting session. We've got some great speakers, 5 great speakers, and so I hope you'll all enjoy it, and I hope you'll all be active in the Q&A at the end of each talk, okay? So first off, it's my great pleasure to introduce Paul Kenney from the Icahn School of Medicine at Mount Sinai. I've known Paul for many years, and he's gone above and beyond by arriving just after midnight this morning due to delayed flights due to bad weather in the US. So thank you, Paul, and hopefully we can keep him awake for the next 40 minutes at least. So Paul is the Ward Coleman Professor and Chair in the Department of Neuroscience, and he's also the Director of the Drug Discovery Institute at Mount Sinai. He trained in Ireland, as you'll be able to tell as soon as he opens his voice, and then he went and did a postdoc at Scripps, and I got to know Paul basically when he was at Scripps, and we've known each other ever since, and I'm really looking forward to hearing all about the development of novel GPCR therapeutics for substance use disorders. Thank you, Paul.
Thank you, Andy. It's great to be here. My hands are a little shaky. I got my coffee, so I hope to stay awake during my own talk. So I'm let's see if this will work here. I'm going to talk to you today about a drug discovery program that's currently active in my lab that's seeking to develop orexin-1 receptor antagonists for the treatment of substance use disorders. Okay, so what is the orexin system? I'm sure most people here are familiar with it, I've heard of it, but for those of you who haven't, orexin, or otherwise known as hypocretin, is a peptide that's produced by just a few thousand neurons around the lateral hypothalamus. The name hypocretin probably gives the name away. It's an incretin-like neuropeptide, and it's produced in the hypothalamus, hence the name hypocretin. That's how it was named when it was discovered by Eloise and colleagues at Scripps. And I'd worked with Eloise at Scripps, and hence my interest in this neuropeptide. But it was co-discovered by another group, and the peptide was shown to regulate consumatory behaviors, feeding behaviors, and hence its name orexis from appetite, or orexin from orexis. And so the behaviors that orexin neurotransmission has been most implicated in is regulation of sleep-wake cycles, and also regulation of arousal. And these actions, of course, are thought to reflect the circuit-based mechanisms of orexin transmission in the brain. So the orexin neurons in lateral hypothalamus project to regions that are involved in sleep-wake transitions, hence that action. And mutations in the orexin neurons, or the orexin peptide, and the receptors that regulate the action in the hypothalamus are thought to be the primary cause of narcolepsy. But orexin neurons also project to regions of the brain involved in arousal and motivation. For example, the hindbrain or the midbrain dopamine system, the ventral tegmental area, and other mid and hindbrain sites involved in arousal, motivation, risk or threat detection and avoidance, for example, or in addition. And so the orexin system is generally thought to regulate this kind of gradation of being unconscious, where you then move from an unconscious sleep state into a conscious state. And that's thought to be regulated primarily by the orexin-2 receptor. And then once you're awake or conscious, varying degrees of arousal, depending on what's happening in the environment, is thought to be regulated by the orexin-1 receptor. So you get this nice division of labor, if you will, where the orexin peptide, or peptides, can act through these 2 receptors. And like I said, the orexin-2 receptor is thought to regulate sleep-wake transitions. So disruption in this arm is thought to be the primary cause of narcolepsy. So if this system doesn't work, sleep-wake transitions are disrupted. And then whereas orexin peptide acting through the orexin-1 receptor is not thought to play a major role in sleep-wake transitions. So mutations in the orexin-1 receptor or pharmacological blockade, as you'll see in a few minutes, of the orexin-1 receptor does not promote sleep, but instead seems to regulate arousal states in awake humans and animals. And so I've been interested in the role of orexin transmission in arousal states, motivation states for many years, particularly in the context of drug addiction, back from when I was a postdoc at the Scripps Research Institute when I was working with Eloise. And so this is actually, I think, the first paper I published in my own lab when I set up my own lab at the Scripps Research Institute. And what we've been looking at as part of this project is whether orexin transmission plays a role in regulating the motivational properties of nicotine, which of course is the major addictive component of tobacco smoke. And so those of you who are not familiar with self-administration procedures, these are all self-administration data here in rats. What we do is implant a catheter into the jugular vein of a rat, and there's an exit port in the animal's back. And that's a chronic implant, so once we perform the surgery, the animals can recover and they're perfectly healthy, perfectly happy, and we can perform these self-administration type experiments for many months, even up to a year, typically in our rats, if the catheter remains patent. And so in these experiments, animals can basically consume their own nicotine. They can lever press or nose poke to earn infusions, and they can choose to do that or not. And if they choose to do that, they can regulate their own, titrate their own consumption. And what we found is in animals that were self-administering nicotine, if we treated them with this pharmacological tool, it's considered a selective orexin-1 receptor antagonist, we got nice dose-dependent reduction of self-administration behavior. So if we disrupt orexin-1 receptor-mediated transmission, the motivation of an animal to earn nicotine infusions is decreased. By contrast, if we have rats that are responding for food pellets, so these are hungry animals, they're responding using the same response criteria as those animals that are responding for nicotine. And if we block orexin transmission, orexin-1 receptor-mediated transmission, we see no effect on food responding. Now, the way we typically interpret these data is not necessarily in the context of appetite regulation, but rather in terms of the ability of the animal to perform the behavioral task. So the assumption here is that these animals are very hungry, they're willing to respond vigorously for food, and what we see is that when we treat the animals with doses of this orexin-1 receptor antagonist that will decrease nicotine intake, it doesn't disrupt the ability of the animal to earn food reinforcers. So we interpret these data as suggesting that orexin-1 receptor transmission is regulating the motivation to earn nicotine infusions, but not by a mechanism that's secondary to just generalized disruption in behavior. So it's not as if the animals are falling asleep here. They're able to perform the task, they just choose not to when, in terms of earning nicotine infusions, when orexin-1 receptors have been blocked. So that's evidence that orexin transmission plays a role in regulating the motivational properties of nicotine. These data here, you'll see why I'm showing these hopefully in a few minutes. This is a different flavor of self-administration, if you will. What we're using here is called a fixed ratio 5 schedule of reinforcement. And all that means is an animal has to press a lever 5 times to earn a nicotine infusion. So it's a directed behavior. In a progressive ratio schedule of reinforcement, each time an animal earns a nicotine infusion, we make it harder for the animal to earn the next infusion, and so on. So eventually the animal has to press up to hundreds of times for each infusion of nicotine. And so progressive ratio schedules of reinforcement are considered good measures of the motivation of the animal to earn a drug. And hopefully what you can see is that the selective orexin-1 receptor antagonist really is quite striking, really dramatic effects on nicotine responding under a PR, progressive ratio schedule reinforcement. And what I'm not showing here is that we don't see any effects on food responding under a PR schedule as well. So again, this is quite compelling pharmacological evidence that orexin-1 receptors regulate the motivation to earn nicotine infusions. But of course, the compound we used is a compound, which means there's no guarantee that it's entirely selective and that the actions that we're seeing when we treat our rats are exclusively mediated by orexin-1 receptors. So to confirm that orexin-1 receptors are indeed regulating the motivational properties of nicotine, we turned to using mice instead of rats. And so we performed self-administration, the very same type of self-administration experiment. But in this case, we used either wild-type mice or mice that have 2 null both copies of the orexin-1 receptors carry null mutations, or we have heterozygous mice where they've got 1 wild-type allele and one mutant allele, so they carry about half the complement of orexin receptor. In other words, wild-type animals, about half the receptors, orexin receptors, are missing from the brain, and there are no orexin-1 receptors in the brain. And what you see is a nice gene dosage effect on self-administration behavior. So again, these are animals self-administering nicotine. These are different doses of nicotine. These are the number of infusions that the animals earn. And in wild-type animals shown in gray, you get your classic inverted U-shaped dose-response curve. So the animals like nicotine, they're responding vigorously for it, and their behavior is sensitive to the dose of nicotine that's available. In animals that got about half the complement of orexin receptors, you can see there's a suppression of responding. And animals that don't have orexin receptors, you can see essentially a flattening of responding. And you may say, well, even in the knockouts, there's residual levels of responding. They're earning about 4 or 5 infusions, so it doesn't look like orexin receptors are regulating all of the motivational properties of nicotine. But actually, if you just simply remove nicotine and replace nicotine with saline, and only saline is available to the animal, you'll see exactly the same behavior. So even when saline is available, there's still a residual level of responding, presumably because the animal is looking for the drug. So what we see in the orexin-1 receptor knockouts is behavior that's indistinguishable from what we would see in a wild-type animal if we substituted nicotine infusions with saline infusions. So again, pretty compelling evidence, in this case behavioral genetics evidence, that orexin-1 receptors regulate the motivational properties of nicotine. And in these same animals responding for food reinforcers under the same fixed ratio 5 schedule, you can see that there's no effect of knocking out orexin-1 receptors. So this looks to be a very behaviorally specific event. Another piece of data, so we were curious as to why. What is it exactly that orexin receptors are doing such that they're able to regulate the willingness of the animal to earn nicotine infusions? And of course, the reason why animals earn nicotine infusions is thought to be because the drug is rewarding. It stimulates and enhances brain reward activity. It's pleasurable. And so the animal consumes the drug because it's pleasurable. In the same way an animal would consume cocaine or opioids because those drugs are able to stimulate or enhance brain reward activity. So we can actually measure the stimulatory effects of drugs of abuse on brain reward or brain pleasure circuits, if you will, using a behavioral procedure called intracranial self-stimulation. So I won't go into the nuances of this procedure, you'll just have to take my word for it. So the idea is that when a drug of abuse like nicotine or opioids, for example, stimulate brain reward systems, that facilitates this ICSS behavior. And so lowering of ICSS thresholds, in other words, an increase in sensitivity to ICSS stimulation, is considered pretty much a direct measure of the reward-enhancing properties of a drug of abuse. And so hopefully what you can see here is that in animals that were treated with nicotine, shown in blue, this is a saline injection, a vehicle injection. These are animals treated with nicotine. There's a lowering of the ICSS threshold. And that's interpreted as an enhancement of brain reward activity, a direct measure, if you will, of the pleasurable, the addictive properties of nicotine. And so what we find is when we treat our animals with increasing doses of this orexin-1 receptor antagonist, those same doses that decrease nicotine self-administration behavior, what we see is an attenuation and then ultimately a complete blockade of the facilitatory effects of nicotine on brain reward circuits. So that suggests that nicotine requires orexin transmission in order to stimulate brain reward circuits. So the rewarding properties of nicotine require orexin transmission. That being the case, it makes sense that animals would stop self-administering nicotine if we block orexin transmission and thereby block the stimulatory effects of nicotine on brain reward/pleasure centers. So that's how we think, or that's how we taught orexin receptor antagonists were working in order to control nicotine self-administration behavior. However, we collected some data in the lab that was initially a bit of a surprise to us, but actually in the literature began to make sense. So we asked how exactly is it that orexin transmission is able to regulate the stimulatory effects of nicotine on brain reward circuits. My presumption was that nicotine can directly stimulate these circuits because of locally expressed nicotinic receptors. Why would you need an intervening neuropeptide like orexin in order for nicotine to actually work on these brain reward circuits. So we were curious to see when orexin transmission was enhanced, when it was being engaged in the brains of our animals while they were performing either nicotine self-administration behavior or, for comparison, food responding. And so we used fiber photometry in hypercretin 1 or hypercretin Cre expressing animals. So we expressed GCAMP, this genetically encoded calcium reporter, in these orexin neurons in the hypothalamus. And then we used fiber photometry in order to record orexin neuron activity as animals engage in either food-seeking or nicotine-seeking behavior. And what we found is when animals were seeking food, there was no change in orexin receptor, orexin neuron activity, as far as we could tell, under either FR1 or FR5 schedules. And so what I'd shown you previously is we block orexin transmission in animals responding for food under an FR5 schedule. There's no change in behavior. So these data are kind of consistent with that. It doesn't look like in animals that are responding for food, hungry animals, that orexin transmission plays a particularly important role, at least as reflected by the data we collected in self-administering animals or in terms of the fiber photometry response. However, in animals that were responding for nicotine, what we see is that there's quite a striking increase in the activity of these orexin neurons. But what was unexpected to us is that when we recorded activity under a fixed ratio of 5 schedule, which is what we typically use for all our experiments, we see a beautiful engagement of orexin receptor activity. However, if we performed the same experiment and allowed animals to receive or to obtain nicotine under a fixed ratio 1 schedule, which is simply just an easier schedule, the barrier to earn an infusion is lower because the animals just need to press the lever once instead of 5 times, we see there's no engagement of orexin transmission. So what that suggests is that orexin transmission does play a role in perhaps regulating the motivational properties of nicotine, but only when the animal has to invest real effort to obtain the drug. At least as reflected by these descriptive fiber photometry data, we see engagement of orexin transmission only when the animal has to forego other opportunities, other things it's doing in the environment to really invest effort to obtain nicotine infusions. So we looked to see if that was actually the case. Is this why orexin transmission is regulating the nicotine self-administration behavior? It's not necessarily regulating the stimulatory effects of nicotine on these circuits, but somehow it's being recruited when an animal has to really focus its efforts on obtaining the drug, not necessarily, as I say, regulating the direct stimulatory effects of nicotine on these reward circuits. So what we did is set up another self-administration experiment. We had animals lever pressing under an FR5 schedule once again, and we treated the animals with this orexin receptor antagonist, this tool antagonist that we've been using. Again, we see a beautiful reduction in self-administration behavior. However, if we lowered the burden, if you will, for animals to earn nicotine infusions, so it's essentially the same behavior, but it's just easier. Animals just need to press a lever once. What we find is that blockade of orexin receptor transmission has really no effect whatsoever on the self-administration of nicotine. So it suggests that orexin is needed. You need to recruit orexin transmission at orexin 1 receptors in order for an animal to persist in its drug-seeking behavior when it gets harder. We see exactly the same thing in our orexin 1 receptor knockout mice. If you remember, I showed you that the full mutant animals had lower levels of self-administration behavior relative to wild-type animals. So we repeated that experiment, saw exactly the same thing. Here are wild-type animals in gray. Here are the knockout animals. You can see that the knockout animals they earn lower numbers of nicotine infusions. That's under the same fixed ratio of 5 schedule I showed you before. If we lower the response requirements, make it easier for animals to earn nicotine infusions, what we see is over the subsequent 3 sessions under an FR1 schedule, an easy schedule, there's absolutely no difference in responding between wild-type and knockout animals. So again, it suggests that orexin transmission is engaged when animals have to kind of forego other opportunities, have to invest effort to obtain the drug. And this, to me at least, is exciting because ultimately that's what we think drug addiction likely is. It's not that you become sensitized to the rewarding effects of a drug, it's that the drug gains more and more importance to the addicted individual such that they invest more and more effort in obtaining the drug at the expense of other opportunities to enjoy non-drug rewards in the environment. It's that corruption of motivational properties or the motivational value of the drug relative to other non-drug reinforcers that really is at the heart of addiction. And so what we're seeing here is a neuropeptide that seems to regulate that very process. When an animal has to work hard and really invest effort to obtain the drug, that's really when orexin transmission kicks into gear. But I showed you some data using the ICSS procedure that suggests that actually the rewarding effects of nicotine are regulated by this neuropeptide. So how does that make sense? If I'm telling you that self-administration behavior is effort dependent, I'd shown you this ICSS procedure that didn't appear to be effort dependent. It just looked like the ability of nicotine to stimulate reward circuits required orexin transmission. So we went back and looked at that behavioral procedure, and it turns out it is actually quite effortful. There's very long delays, inter-trial interval delays, timeout delays. The animal has to be very patient throughout the ICSS procedure. So we decided to look what would happen if we made that procedure just easier. And so we designed a new procedure. I won't go into the nuances here, but basically we had our standard ICSS procedure or a modified procedure with markedly shortened inter-trial intervals and timeout periods so the animal could just breeze along in this procedure without having to wait and invest effort. And what we found is under the standard procedure, once again, we replicated our effect where nicotine will stimulate brain reward circuits, and that stimulatory effect on these reward circuits is reflected by lowering of ICSS thresholds. It's completely abolished when we pharmacologically block orexin-1 receptors. However, when we modify the ICSS procedure to make it easier, substantially easier, what we find is that nicotine, once again, will stimulate brain reward circuits as measured by lowered ICSS thresholds. But in this simplified procedure, there's absolutely no effect of blocking orexin-1 receptors. So we think that we totally misinterpreted our findings, that orexin-1 receptors are not regulating the stimulatory effects of nicotine on brain reward circuits. Orexin-1 receptor transmission is important for the animal to remain engaged with the behavioral procedure. You treat the animal with nicotine, that facilitates this process, but that engagement requires orexin-1 mediated transmission. So again, we think that this is not just a case of orexin-1 receptors are involved in reward-related behaviors per se, but it's much more nuanced, if you will. It's involved in the engagement of the animal, the motivation of the animal to persist in either drug-seeking behaviors or to persist in a behavior where it experience the pharmacological actions of the drug. And again, from a therapeutic perspective, from my perspective, I think that's far more interesting. We're not looking to simply block the rewarding effects of the drug and hope that that will translate into decreased drug-seeking behavior. Orexin-1 receptors appear to be regulating that investment of effort, if you will, the value of the drug that the animal places on obtaining the drug, which again is what we think is at the heart of drug addiction. Okay, so how is all this working? How is orexin regulating the motivational properties of the drug? And if you were to look in the literature, the takeaway would likely be, or your presumption would likely be, that orexin receptors are somehow regulating the stimulatory effects of nicotine and other drugs of use in midbrain dopamine systems. There's a number of papers been published suggesting that orexin receptor antagonists will block the stimulatory effects of nicotine, cocaine, morphine on VTA dopamine neurons and thereby attenuate or decrease drug-induced increases in dopamine transmission in the nucleus accumbens. So this VTA to accumbens circuit, this mesolimbic dopamine circuit, is really thought to be critical for the motivational properties of drugs abuse. So of course the presumption is you block orexin transmission, you'll block the stimulatory effects of nicotine on VTA nucleus accumbens circuits. So our idea was this should be a pretty simple explanation. Orexin-1 receptors are somehow regulating the stimulatory effects of nicotine on the VTA nucleus accumbens circuit. And so we sought to test that hypothesis. So the first thing we did was to get a group of animals, a group of rats, have them self-administer nicotine, and then we simply infused this same orexin receptor antagonist I've shown you before directly into the ventral tegmental area. And so we've done this in 3 separate cohorts of rats, and what we find is there's absolutely no effect of intra-VTA infusion of this antagonist on self-administration behavior. And hopefully what you can see here is that the dose of nicotine, or of this antagonist that we use, 1 microgram per 0.5 microliters, is about 1 million-fold above the IC50 for this antagonist at orexin-1 receptors. And it's orders of magnitude higher than the amount of this antagonist that would make it into the VTA after systemic injection. So to me, there's absolutely no way systemically administered orexin receptor antagonists are acting in the VTA to decrease self-administration behavior. And so we've done this, as I say, in multiple cohorts of animals. We're quite confident in this finding. But we also looked in our knockout mice. If you remember, the orexin-1 receptor knockout mice self-administered far less nicotine than wild-type animals. We took the missing orexin-1 receptor, we cloned it into a lentivirus. We confirmed that we got good expression, or re-expression, if you will, of the otherwise missing orexin-1 receptor in the brains of the knockout animals. And we functionally rescued orexin-1 receptor expression in the VTA of our knockout mice. And what did we find? Absolutely nothing. So when we restore orexin-1 receptor expression in the VTA of our knockout animals, that's shown here in the dark blue, we saw the same deficit in nicotine responding that we saw in the knockout animals that were treated with a control virus. So between the antagonist, the local antagonist data into the VTA, and the virus rescue experiment, we think that these are compelling data that the notion that orexin-1 receptor antagonists are working in the VTA to somehow disrupt the motivational properties of nicotine is likely not true. So if that is the case, where on earth are these orexin receptor antagonists actually acting in the brain to regulate the addictive properties of nicotine? So we started hunting around by simply infusing the orexin receptor antagonist in various brain regions, candidate brain regions, and really didn't see anything. So we took a step back and actually asked, well, where on earth are these receptors actually expressed at high concentrations in the brain? Because they're likely the sites that are, you would imagine, are involved in regulating the actions of an orexin-1 receptor antagonist. So the first thing we did was to get hold of an orexin-1 receptor YFP reporter mouse, and we simply looked to see which cells fluoresce in the brains of these animals, with YFP being driven under the orexin-1 receptor promoter. And so what we found is various regions where you'd expect to see orexin-1 receptors being expressed that are consistent with previously published receptor autoradiography studies and in situ hybridization studies. So the hypothalamus, the tinea tecta, the locus coeruleus. But what really caught our attention was there was a beautifully circumscribed population of orexin-1 receptor expressing cells located right next door to the habenula. And so this kind of parahabenula region, it's not actually in the habenula, it's located right beside the habenula. And why we're interested in that population of cells as opposed to the others that we were able to detect in the brains of these animals is that my lab had previously published that the habenula plays a key role in regulating not the rewarding effects of nicotine, but the aversive effects of nicotine. So the idea we had is perhaps orexin-1 receptors are not necessarily regulating the reward-related effects of nicotine, but the aversion-related effects of nicotine, such that if you don't have orexin-1 receptors, maybe the aversive properties of the drug are somehow enhanced, and that somehow devalues the drug. So that was the hypothesis. So we focused on this site, and we just wanted to confirm that the orexin-1 receptors are indeed expressed in this part of the brain. After all, that's a YFP reporter mouse, so we've looked at this in a few different ways. This is in situ hybridization using RNA-scope. And what we found is indeed, hopefully you can see in green, the orexin-1 receptor transcripts are in this parahabenula region. Interestingly, those of you who are familiar with the habenula and the thalamus know it's in rodents thought to be exclusively glutamatergic. We know that's the case in primates. It's thought to be almost exclusively glutamatergic. What we found is that's actually not strictly true. There are small pockets of GABAergic cells located, distributed around the intralaminar thalamus, around this boundary layer. It turns out that the orexin receptor in this part of the brain is expressed exclusively by a population of GABAergic neurons located in dorsal thalamus, right next door to the habenula. So already, the cellular biology looked a little interesting. This is a group of cells that we know really nothing about. So we patched onto these cells, and we found using ePhys recordings that they were depolarized in response to local puff applications of orexin peptide. And actually what we found is if we patched on to cells around these orexin-1 receptor cells, we could see an orexin peptide-induced increase in GABAergic transmission. So what that means is these orexin receptors are expressed by GABAergic neurons in this dorsal thalamic region. And if we stimulate those cells using orexin peptide, we increase local GABAergic transmission in the thalamus. And it turns out there were only certain cells that were responsive to this event that would show an increase in GABA transmission. And we call these cells type B cells. They're actually pretty interesting. They've got different electrophysiological characteristics and morphological characteristics than other cells that surround them in the thalamus. And this is something that we're really interested in. We're exploring in terms of the projection patterns of these cells and what happens if we directly manipulate them. But anyhow, the take-home message here is that we have an area of the brain that seems to express these functional orexin-1 receptors, and it's a part of the brain that we thought may play a role in regulating the motivational properties of nicotine. Anyway, so we decided to directly test that. So we used the same two approaches I've already shown you. We took a group of animals that were self-administering nicotine. We infused the orexin receptor antagonist that I've shown you before directly into this brain region, using the same concentration that didn't work in the VTA. And in this case, when we infused it directly into this parahabenular region, there was a striking reduction in self-administration behavior, very reproducible. So it looks like this is a functional population of orexin-1 receptors that may regulate what we see when we deliver this drug systemically. So we went back to our knockout mice, and then again, we injected the animals with either a control lentivirus or a virus that will re-express the otherwise missing orexin-1 receptor. And what we found again is in the knockout animals treated with control virus, they demonstrated a striking reduction in their willingness to earn nicotine infusions. However, when we rescued orexin-1 receptor expression locally in this parahabenular region, the knockout animals now looked indistinguishable in terms of their responding for nicotine from wild-type animals. So it suggests that this population is indeed important in regulating the actions of orexin receptor antagonists. And indeed, on data I'm not showing you, we took these very same animals and injected them with the SB compound, and we saw no effect in the knockouts. And then in the knockouts with rescued virus, they were as sensitive to the orexin receptor antagonists as the wild-type animals. So this population in this parahabenular region of the brain that we know next to nothing about looks to be an important population that regulates the anti-addiction effects, if you will, of orexin receptor antagonists. I don't have time to tell you how these are working. One thing I will say is that our hypothesis that this population was somehow regulating the aversive properties of nicotine is completely wrong. This site does not look like it has got anything to do with the habenula. Instead, it appears to regulate corticothalamic and thalamocortical transmission. So presumably, those corticothalamic interaction events are important in value allocation. That's our working hypothesis, something that we're looking at in the lab at the moment. Anyway, you'll just have to take my word for it that this area of the brain looks like it's regulating the motivational properties of nicotine, but not by regulating the basic reward or aversion effects. Oh, okay. So I'm running out of time, so I'll cut to the chase. So we've taken this data, and on the back of that, we thought, wouldn't it be nice to be able to develop an orexin-1 receptor antagonist that we'd actually advance into human clinical trials for the treatment of tobacco dependence and other substance use disorders. So we went to the patent literature, and we actually found a whole cluster of antagonists that were designed as mixed OX1, OX2 antagonists, but those that had OX1 activity had a common pharmacophore, and it's shown here, it's in dark. So we took advantage of that. We set up a medicinal chemistry campaign. This is with our collaborator, Ted Kameneka at Scripps, Florida, and we sought to develop a drug-like orexin-1 receptor antagonist. We did, after a number of years, identify a drug-like, very selective, potent selective orexin-1 receptor antagonist, or a series of compounds we call the 9659 series. And from that, we developed a compound derived from the series called AZD4041. It's called AZD because we actually partnered with AstraZeneca in this program. I'm sorry AstraZeneca won't let me present the structure, hence it's grayed out. But the compound looks just beautiful from a physiochemical and pharmacological perspective. Very potent, very selective. It's got drug-like physiochemical properties, it's brain penetrant, and it's got what you'd like to see in a drug, hopefully with therapeutic actions. We have confirmed that this compound is active in our rats. It will dose-dependently decrease self-administration behavior. If we dose every day, we don't see any evidence of tolerance. The animals don't demonstrate any evidence of sleep-promoting effects or responses to the antagonist, and they don't demonstrate any non-selective behavioral abnormalities that could explain the effects. It looks like it is specific to the disruption of the motivational properties of nicotine. And we've also tested the compound in non-human primates, and again, we got beautiful reduction in self-administration behavior with no effect on food responding. The compound has moved through, we actually got an IND approved by the FDA. It has moved through phase 1 MAD and SAD studies, and currently it's primed, if you will, to start efficacy testing later this year. So we have our fingers crossed that we'll see something in a human efficacy study. And I'd like to finish by thanking everyone in my lab and at AstraZeneca and the NIH who are involved in in these experiments. Thank you.
Great, thanks very much, Paul. Because of time, we've got time for one question, so Danny, go. So if anyone else has a question, please, can you wait till afterwards and speak to Paul afterwards, because we've got to keep to time. But you go, Danny.
Hi, Paul. You relieve some anxiety because people... Is that working? Yes. So you relieve some anxiety because people try to develop some orexin receptor agonist, and we're always afraid that these compounds will induce addiction and things like this. Now, although most of the stuff is on orexin 2, it's even better to know that orexin 1 may not have all these bad properties that we expected.
Yeah, it's lovely to see you, Danny. I'm sorry I went over in time.
No, it's fine.
I will just quickly respond to that. We've also been trying to develop orexin 1 and orexin 2 receptor agonists also, and we have not seen any evidence that they've got a pro-addiction reward-related effects. It looks like it's very much related to motivation, recruitment of behavioral resources. So I don't think they're going to have pro-addiction effects. Thank you.
Okay. Stay there, Paul. I'm going to make an executive decision because it's only lunch after us, so we're going to keep going. Please, have your question.
Okay, I had 3 questions, so I just go for one of them. So I assume that the data you showed us is about adult male mice, right?
Adult animals in all cases, yes.
What about females? Because orexin effects is already reported is highly sex related.
Yes, so what about sex? We see that in female knockout mice and in female rats treated with orexin receptor antagonists, the effects are pretty much indistinguishable from males, but the pharmacokinetics of any of the antagonists that we've dosed are very much sex dependent. So we've published a paper a number of years ago at RON-C showing that the estrous cycle in female rats actually influences the rate of metabolism of the SB compound I'd shown and some other receptor antagonists. And actually, sex-dependent PK properties are surprisingly not taken into consideration too often. So that's something we're very much mindful of as we now have advanced to human clinical studies. And we are actually looking at the PK properties of our compounds in male and female subjects. So.
Okay, over here, please.
Hi, thank you. Very good talk, Sal.
Yep.
So I was just wondering, orexin receptor antagonists have been on the market for insomnia for quite a long time. Is there any evidence of these showing anti-addictive effects?
Actually, yes. There was a study published in Science Translational Medicine about 18 months ago by Kelly Dunn's group. And one of the major, disturbances, symptoms that you see in people who are opioid addicted, OUD patients, is, of course, the drug is a narcotic, so it disrupts sleep. And so those patients have really disrupted sleep-wake cycles. So because of that, they tested the effects of suvorexant in OUD patients to see if it would kind of help alleviate some of the sleep disturbances that you see. And indeed it does. One of the serendipitous findings of that paper, report in that paper, is it beautifully attenuates craving for opioids. And in fact, the paper hypothesizes that the sleep-promoting effects are because of orexin 2, whereas the anti-craving effects may actually be the orexin 1-related actions of suvorexant. So that was just a beautiful study, and we were delighted to see some POC data, if you will, in humans.
Just to extend on that, I actually had a trial of suvorexant in comorbid alcohol use disorder and insomnia, and Merck pulled the pin on it because we got the pandemic and the lockdowns in Melbourne just destroyed us. But when we unblinded the data, we've got, under review at the moment, there's a case study where a guy came in mid-30s, comorbid insomnia and AUD, 13 weeks later, 100% abstinent, no more insomnia, his liver was working properly. So we just need more trials on it. So, but I think, as Paul's saying, the selective OX1 antagonist from an addiction perspective is probably a better long-term option.
That's the current thinking. We'll see.
But if you've got comorbid insomnia, then suvorexant might be good. And a lot of people with AUD do have comorbid insomnia, as you know.
Yes.
Anyway, we better call it a day here. So thanks very much indeed, Paul. Thank you.
Thank you.
Okay, our next speaker is Dr. Leigh Walker from the Florey Institute of Neuroscience and Mental Health in Melbourne. Leigh is an HMRC fellow and has a lot of research interest in the neurobiology of anxiety and alcohol use disorders. And today she's going to speak about some of her work looking at muscarinic acetylcholine receptors in the context of AUD. Leigh.
Thank you very much. thank you for the introduction and for the organisers for having me here today. I am looking forward to talking about this work, which has been done in collaboration with both of our co-chairs. So I'm going to be talking about muscarinic acetylcholine receptors in alcohol use disorder, and hopefully I can get this all to work. So just a bit of background in terms of alcohol use disorders. We all know that it's a major socioeconomic burden. And it causes, 5.3% of global deaths per year. In Australia, it leads to 430 hospitalisations and 15 deaths daily, and costs the Australian economy $22 billion every year. So I'm sure that that's the same in a lot of countries around the world, or it is the same. But one of the big problems is that the current treatment options that we have for alcohol use disorders are really inadequate at a population level. So they suffer from limited efficacy, adverse side effects, and low compliance. And there's been no new FDA-approved medications for alcohol use disorders for almost 20 years now. And a big problem with this is this translation hurdle. So what we're doing in preclinical models, so there's been a number of targets that have been identified in preclinical models, but there's really been this hurdle to translate that into the clinic, and what we're really missing. And even if we do get them into clinical trial, most of these compounds don't make it through clinical trial. So 9 out of 10 won't make it into the clinic from there. And so when we were really thinking about this issue, we were thinking about the way that addiction is characterized in this sort of shift from controlled to compulsive and inflexible alcohol and or drug seeking. And this is really thought to be regulated by the striatum in the brain, the caudate putamen, and the shift is thought to be regulated by movement and activity from the ventral striatum, the nucleus accumbens, up into the dorsal striatum or the caudate putamen. So from light to heavy drinkers, sorry. And this has been shown in humans with bold studies. Sorry, I lost my mouse. With light or social drinkers having activity in the ventral striatum, whereas you look in heavy drinkers and you see activity in the dorsal striatum. And this also correlates with their obsessive alcohol seeking score as well, the activity in the dorsal striatum. And not only is there a shift from ventral to dorsal engagement of the brain, but also this medial to lateral shift. So this is data from rats where they did a devaluation, so you have a rat pressing a lever to get alcohol, but before the test they devalue the alcohol. So they can either devalue with a different reward, so in this case it was sucrose, so that would be the non-devalued, or devalue the alcohol specifically, just give them access to alcohol. And what you can see is that after 8 weeks of responding for alcohol, the rats sort of show this habitual responding, whereby they respond despite having access to alcohol before. But if you sort of inhibit the dorsal lateral striatum, but not the dorsal medial striatum, you can sort of renew this sensitivity to devaluation, suggesting that the dorsal lateral striatum is key in this habitual responding for alcohol. And so to probe this, we took a sort of a reverse translation response. We started with human tissue from the NIAAA sponsored New South Wales Brain Bank of caudate and putamen, and we took tissue from 10 people with alcohol use disorder that were non-smoking, and 10 controls that were age and sex matched. And we did genome-wide RNA sequencing to look at the differentially regulated genes. And what was interesting to us was that the top candidate that came out of this in terms of canonical pathways was G protein coupled receptors. And when we looked closer at these receptors, there were a number that were sort of not a big surprise: dopamine, serotonin receptors, cannabinoid receptors. But what was really interesting to us was the muscarinic, the acetylcholine muscarinic 4 receptor. And so we validated this data using qPCR, where we split out the caudate and putamen, and showed that this reduction in expression was specific to the putamen, or the dorsal lateral striatum. And so then the next thing that we wanted to do was to take this and see if we could replicate it into a rodent model. So to do this, we gave rats access to alcohol for a really long time. So for 6 months, they had intermittent 2-bottle choice. And we had 3 groups. So the first group had just access to alcohol and were sacrificed 24 hours after their last alcohol exposure. Rats that were sacrificed 14 days after their last alcohol exposure, and alcohol naive and age-matched rats. And this is just looking at their stable alcohol consumption. So these were also, inbred alcohol-preferring rats, so they drink a lot of alcohol as well. And what we did was we then punched out the dorsal medial and dorsal lateral striatum, so the corresponding regions from the human, and did qPCR on these. And what we saw was that, in line with our human data, was that we could recapitulate this reduction in the M4 receptor expression after chronic alcohol and abstinence. And we also saw that this was specific to the dorsal lateral striatum. The ventral striatum showed no differences. And so, I mean, obviously, as my talk gave away, so this led us to a lot of interest in muscarinic acetylcholine receptors, which there are 5 different subtypes, M1, M2, M3, M4, M5, so they are a Class A GPCR, and the odd numbers signal through GQ, so are stimulatory, and the even numbers signal GAO and are generally inhibitory in the cell. So there's been quite a bit of evidence using knockout mouse lines and non-selective compounds that have suggested that they have roles in alcohol and drug consumption. And I don't know why this is being so slow, sorry. And not only that, they're expressed widely throughout different parts of the brain that are really important for addiction. So in the striatum, hippocampus, midbrain, and hindbrain areas. And specifically within the striatum, the M4 receptor is known to be expressed on cholinergic interneurons and also on D1 medium spiny direct outputs. And, through this in the striatum and indirectly as well through hippocampal inputs, they're also known to modulate both glutamate and dopamine release in the striatum. And so what we wanted to understand next then is, where is this dysregulation specifically in the dorsolateral striatum? So we used RNA scope to quantify the number of mRNA puncta that were associated with each of these cells. So we counted both the cholinergic interneurons and the D1 MSNs as well, and we quantified what you can see here, the green dots, so sorry for anyone who is colorblind, the M4 receptor expression. And we didn't see any difference in the dorsal medial striatum in line with what we'd seen in both the human and our qPCR, but we saw a specific reduction in M4 receptor expression on the D1 MSNs in the dorsolateral striatum. And so then we wanted to really understand, okay, so we can see this dysregulation, but what does it actually mean? Can we actually use this as a drug target? And so one of the big problems with muscarinic receptors has really been that the orthosteric binding site is really well conserved. So having drugs that specifically target subtypes of muscarinic receptors have really been lagging, and this has really hindered the subtype selective understanding. So our collaborators at Vanderbilt University have these drug development pipelines where they're looking at different allosteric modulators. So instead of turning instead of turning a receptor on or off, that it's modulating the signaling. And so they developed this compound, VU154, which is a positive allosteric modulator for the M4 receptor. And we then took this into rodents to see if we could alter alcohol consumption and seeking. So this is just a video of a rat pressing a lever. So what you'll see is that when the rat presses a lever, a light comes on, a cue light, and then they have the reward delivered into the receptacle. So this is similar to what Paul was just, Paul was just talking about in terms of the IVSA, except for that the alcohol is just delivered into a portal. And so what we can do is we can give the rats drugs and see like how it changes their consumption and seeking behaviors in these models. And so what we saw was that when we administered VU154 that we administered via oral gavage 3 hours before, that we could reduce alcohol consumption at a high dose of 30 mg per kg, and we sort of saw this dose response. And we also saw this in a binge drinking model in mice, so showing that we can see across species as well. And recently we've shown that this is not specific to males. We can also reduce consumption in females in a very similar manner in both paradigms. And of importance, this is specific to alcohol in everything that we've tried. So we've done similar dose responses with sucrose in binge drinking and in operant self-administration. And we see nothing. And we also see nothing in terms of locomotor activity and rotorods, except for at really high doses at 100 mg per kg. And so, as Paul was also explaining, so we can look at not only their consumption, but also their motivation. So it's probably a better measure in terms of addiction. So we can use a progressive ratio where it gets harder for them to receive the reward. And we saw similarly that the VU, the M4 compound, can reduce their reward seeking, their breakpoint, and it also prevents or reduces reinstatement to alcohol, both elicited by a cue and by stress as well. And so, it's gone funny. So we wanted to understand, is this actually happening through the dorsal lateral striatum? So we micro-injected the drug into the dorsal lateral striatum and looked at self-administration and also reinstatement. And unfortunately, you can't see the timelines, but you can see here that this micro-injection reduced both self-administration and also cue-induced reinstatement of alcohol consumption. So suggesting that this brain region is involved. But as you can see, so it doesn't necessarily completely remove the whole effect that we saw. And we've also shown similar things with ML375, which is an M5 NAM. So if we're thinking about, the opposite sort of receptor, modulating it in the opposite sort of way. So now inhibiting a stimulatory receptor. And this work was done by Alice Parisi, who is in the audience today, as part of her PhD when she was a PhD student with Chris and Andy. And she showed that systemically, ML375 could reduce alcohol self-administration without affecting sucrose self-administration, and also reduced cue-induced reinstatement. And again, that this was specifically through the dorsal lateral striatum, but not the dorsal medial striatum. But what about other brain regions, right? So we can see some reduction in the dorsal lateral striatum, but we're not completely eliminating the effect. And so what we did was we went back to our model. So we've sort of made this model of cholinergic dysregulation in a rat. And so can we probe other regions? And two of the regions that we were really interested in are the VTA and the ventral subiculum, so part of the ventral hippocampus. And so we just did qPCR to look at the dysregulation of receptors there. And what we saw was that there was absolutely no difference in the VTA, which was quite surprising to us, based on other work that's shown involvement in other types of reward seeking. But we did see specific changes in the ventral subiculum. So we saw a reduction in M4 receptor expression after abstinence and an increase in M5 receptor expression after alcohol. And so the ventral subiculum, for those who aren't aware, is part of the ventral hippocampus. It's a main output, and it integrates sort of this contextual and spatial information and relays it out to reward outputs, so out to especially a really strong projection to the accumbens shell. And Yavin Shaham's lab has shown a lot of work looking at this disconnection of the ventral subiculum to the accumbens shell and its involvement in context-induced relapse to both heroin and alcohol consumption as well. And so we wanted to see, are these receptors even expressed on this population? So to do that, we did some retrograde tracing with RNA-scope and showed that the M4 and the M5 receptor are expressed on these ventral subiculum projections that are going out to the accumbens shell. And then to test whether they're functionally involved in this brain region, we just did some pharmacology. Where we looked at context-induced reinstatement, and we saw that the M4 PAM was able to reduce context-induced reinstatement, but had no effect on self-administration when micro-injected into the same brain region, whereas the M5 NAM had no effect on context-induced reinstatement, but had a small but significant effect in the first 15 minutes of the progressive ratio. So maybe suggesting that it's reducing the really initial motivation to consume alcohol. And this is really interesting as it aligns sort of with what we'd seen in terms of the cholinergic dysregulation. So this down-regulation of the M4 receptor, if we restore that back up during the abstinence phase, which more aligns with the reinstatement because they've gone through an extinction phase where they have no access to alcohol, whereas the self-administration where they're having daily access, and we see an increase in the M5 receptor, and if we can tone that back down, then we see a slight reduction as well. And I'll just talk very briefly about the M1 receptor. So this one has also been primarily linked to more cocaine and other substance abuses. And so we wanted to see whether it's involved in alcohol consumption. We use this M1 agopam, a Pfizer compound. And again, sorry, this isn't working, but we did see reductions in alcohol consumption, but we also saw reductions in sucrose consumption and both water and food consumption. So sort of suggesting that there seemed to be a non-specific reduction in consumatory behavior. And so we haven't really gone any further with this compound, but there are other compounds developed by Vanderbilt as well that are in that look a lot more promising for cocaine and other stimulant use disorders. So I'll just sum up there. So what I hope I've shown you today is that we have this translationally relevant animal model that we can sort of recapitulate this cholinergic dysregulation that we see in humans back into our rodents. And in terms of our animal models, we've shown that the M1 and agopam reduced alcohol consumption, but also other consumatory behaviours. And we're not really sure if this is the drug that we've chosen. There's quite a lot of debate about whether pure agonists, pure PAMs, agopams, what's the best way to go in terms of M1 receptors. But in terms of what we've seen, it seems to be non-specific. In terms of the M4, we see that it reduces alcohol consumption, motivation, and seeking. And we really see that the biggest effects are when there is a motivational drive or a seeking or a craving involved as well. And we've shown that the dorsal striatum is involved in all aspects, the consumption, motivation, and seeking, and the ventral subiculum is also involved in seeking. But we're not ruling out the fact that there are other brain regions also involved. And then in terms of the M5, systemically, we've shown it's involved in alcohol consumption and seeking. In the dorsal striatum, we've shown that it's involved in consumption, but we haven't probed further in terms of a lot of those seeking paradigms there. And then in the V-sub, involved in the initial phases of the motivation. And again, where it's involved elsewhere. And one of the really interesting things that I think has come out of this is the fact that in the ventral subiculum, the M4 and the M5, they're expressed on the same cells there, but seem to be doing completely opposite things. But we're not sure why. But what we are really interested in, and unfortunately I'm not allowed to talk about a lot of the work that we're doing at the moment, is the potential for repurposing these compounds in alcohol use disorder. So there's a number of a bit of interest in terms of therapeutic companies around muscarinic compounds, both agonists and PAMs that are in development primarily for schizophrenia. But we're hoping that, with the approval of these drugs, KarXT being the one that will be approved most likely by the end of the year, around the end of the year, that we can repurpose these into alcohol use disorder. And with that, I'd just like to thank all of the people who have been involved, especially Andy and Chris and Alice, and thank you all for your attention.
Thank you very much, Leigh, for an outstanding talk. The floor is open for some questions. Karen?
Sorry, I'm much shorter than whoever was on this last time. I was just wondering around adverse effects, liability, and some of the maybe potentially peripheral effects of some of the agents you've been testing, if you could comment on that.
Yeah, so we haven't directly looked at that. Majority of the adverse effects around muscarinic receptors are peripherally thought to be through M2, M3. That's why we think that selective compounds that targeting the M4 or the M5 will be better. If you look at KarXT, which is a compound in development by Karuna, what they've done is they've added a peripherally restricted antagonist, trospium, so that they can reduce those. But in terms of the compounds that we're using a tool compound. It actually has very little selectivity at a human muscarinic receptor. So it would need to be developed in terms of the other compounds coming through. But yeah, that's as much as I can tell you. Sorry, Karen.
So I was intrigued by the overlap with the cannabinoid and muscarinic receptors being modulated in the same way. And of course, rimonabant, which obviously got withdrawn from the market, would have been indicated for use disorders as a kind of follow-on. So do you think there's going to be a bit more selectivity about targeting the muscarinic receptor to reduce the potential for suicidal ideation in patients?
Yeah, I don't think that in terms of xanomeline in clinical trials and the other compounds that have gone into clinical trials, I don't think that there's been, the adverse side effects have been limited. In terms of the link between muscarinic and cannabinoid receptors, it's actually a CV2-dependent mechanism that the M4 receptor is working in the striatum to modulate dopamine release. So it's not acting alone, it's an indirect action. So whether it's going to be more selective, it's going to be, but the indirect actions of it, I guess, is kind of a little bit more complicated. Yeah.
A question for you, Leigh. It's obviously very promising. The M4 compounds look very good, and obviously we have compounds in clinical development that could potentially be repurposed. But if you're designing a compound from scratch based on the knowledge that you had, what would it look like? Would you be combining an M4 PAM and an M5 NAM, or what do you think an ideal sort of target product profile would look like in the context of AUD?
Yeah, that's really interesting. I think that the M5 is also expressed on cerebral vasculature, which might be the limitation in its development, and I think that that's why we've been a lot more interested in the M4. I think M4 PAMs are going to be good, but whether there is other drugs that these are interacting with, other combination therapies that these can be used with, I think will be the future of these compounds and for a lot of disorders that we're trying to treat.
Okay, well, if there are no more questions, please join me in thanking Leigh for an excellent presentation. Okay, we're a couple of minutes ahead of schedule, but our next presentation is one selected from abstracts, and it is from Eloise Kuijer, who speaks to us from the University of Bath about analysis of kappa opioid receptor activation in the paraventricular thalamus. And Eloise, the floor is yours.
Let me just see if the pointer works. Like that. Okay, cool. So, hi everyone, my name is Eloise and I'm a final year PhD student at the University of Bath in Chris Beatty's lab. And today I'll be showing some of our data looking at kappa opioid receptors in the paraventricular thalamus. So the opioid family in the central nervous system consists of three main members. We have our mu, delta, and kappa opioid receptor. And even if you're not very well acquainted with the opioid family, you might have come across the mu receptor, because this is where morphine and heroin have their analgesic and rewarding properties. Interestingly, particularly these rewarding properties of mu are counterbalanced by aversive signaling through the kappa receptor, and this is where my project is all about. So we've talked about the receptor, let's talk about the brain. So which parts of the brain do kappa agonists recruit? To find out, I injected a kappa agonist into mice, black 6 mice. An hour later, sliced the brain and did immunofluorescence to count C-false positive nuclei, which is a marker of neural recruitment. And the title of the talk kind of gives it away. But of all these brain regions, one surprising finding was that the paraventricular thalamus, both the anterior and the medial part, roughly doubled their amounts of recruited neurons, which made us very interested into this brain region. So what does the PVT do? It is thought to integrate homeostatic stress with reward-related information. So it gets a lot of information of the lateral hypothalamus, and as in Paul's talk, this is orexigenic input. And it seems to integrate this with higher mesocortical limbic systems, such as the nucleus accumbens, prefrontal cortex, and hippocampus. Because it sits at the interface of stress and reward, it has now become appreciated that it might actually play a very important role in stress and reward-related psychopathologies, such as depression, but to interest of all of us in this room right now, drug addiction. But what is known about the kappa receptor in this brain region? Not a whole much. We know that overall there are a lot of kappa receptors, and there is a lot of this endogenous ligand dynorphin, but exactly how this works on a single neuron level is less well understood. And this is what my talk is all about. So we can all summarize this as, what I'm trying to do is map out kappa signaling in the PVT. We're interested in, like, where are they, what do they do, are there differences between ages and sexes, and are there co-expression patterns of paraventricular thalamic neurons that express functional kappa receptors with other GPCRs? And furthermore, try to expand this a bit more, looking at kappa signaling on presynaptic terminals. And the way to do this, to get that single neuron data, is using whole cell patch clamp. So, for the non-electrophysiologist in the room, whole cell patch clamp is a technique. You have an electrode in a glass pipette, you get really close to a cell, you break the membrane, and now you have electrical contact with a single neuron that you hope is still alive and kicking. And now, because the kappa opioid receptor is coupled to these potassium channels, if it binds to its agonist, it will open these potassium channels, and this current of ions will flow out of the cell. And because we effectively have an electrode stuck in this cell, this is something we can measure. And it looks a bit like this. So this is a trace. So here the cell is doing its thing. We add spiraline, which is a very selective kappa agonist. The channels open, potassium starts flowing, and the trace goes up, and it stabilizes after a while. And then we co-apply NorBNI, a very selective kappa antagonist. The channels close, and cell goes back to baseline, confirming that this is a very selective kappa-induced current. And now with these currents, there's two main things that we're interested in. One, do PVT neurons produce such currents? Yes or no? Do they have the functional kappa receptors? And B, if they do have the current, if they do produce a current, how big is this current? Doing this a whole bunch of times, we find that 74% of PVT neurons produce such a current. When we look at where these currents are, what is interesting is that particularly in the middle part of the PVT, the size of the current is smaller compared to the anterior part, so towards the nose of the mouse. There is a bit of discussion in the PVT field exactly what is anterior, medial, and posterior, where do we draw the boundaries? So in C, I just plotted it out based on millimeters from Bregma, and also here it is U-shaped. Furthermore, we see no differences in expression or size of current between males and females, or 4-week-old animals and 8-week-old animals. So we've talked about the kappa receptor, which mediates stress and aversion. But what do we find about the mu receptor, which does hedonia and reward? To our surprise, almost all neurons in the PVT that express functional kappa receptors also express functional mu receptors. And this was quite surprising. And there seems to be something going on because the kappa current is always the same or smaller than the mu current. I have not patched a single cell where the kappa current was bigger than the mu current. Exactly what this means and how kappa and mu, how these receptors interact on single neurons is something that we are currently working on. So as a little interim recap, we've previously discussed postsynaptic effects. So this is looking at kappa receptors that are situated on the neuron that we're patching. But the brain's not just one neuron, it's a bunch of neurons. They talk to each other using neurotransmitters. So what can we find about kappa receptors on these presynaptic terminals? So to illustrate, here we have a comically large synapse that is synapsing onto the neuron that we're patching. This is a glutamatergic excitatory neuron, and it's filled with vesicles that contain glutamate, the main excitatory neurotransmitter in the brain. And these vesicles will just be released onto the cell that we're patching, and we can visualize these like these little blips that go down, or EPSCs in the formal terminology. And now, because of the type of receptor that the kappa opioid receptor is, if there are kappa receptors at this presynaptic site, they will inhibit the frequency of these blips. It would decrease. And this is exactly what we see. So here in this graph, I put the baseline to 1, and then of the baseline frequencies, and then when we apply dynorphin, the endogenous ligand of the kappa receptor, we see that the frequency of these blips roughly halves. And then when I co-apply NORBNI, it goes back to baseline. So yes, we have kappa receptors on presynaptic glutamatergic nerve terminals, and here they decrease neurotransmitter release. Now, there's not only excitatory neurotransmission going on, there's also inhibitory neurotransmission going on. The idea is pretty much the same, but now the vesicles contain GABA instead of glutamate. The blips go up because they are inhibitory. But we see pretty much the same. When I apply dynorphin, the frequency of this neurotransmitter release roughly halves. And it goes back up a bit with NORBNI. So to conclude, the PVT integrates stress with reward-related information, but how it does it on a mechanistic level is completely unknown. But we report that 74% of single PVT neurons express both the kappa and the mu receptor, so they express both this stress and aversive-related receptor and the reward-related receptor. Functionally, we also find kappa receptors on presynaptic sites where they both decrease glutamatergic and GABAergic neurotransmitter. And lastly, systemic kappa agonism recruits the PVT. So then I would like to thank my supervisory team, my lab mates, and the British Pharmacological Society for a travel bursary that allows me to present here to you today. And for all the lovers of drug-seeking behavior and behavior like that, I also have a poster looking at kappa agonist effects on heroin-seeking behavior. And if there's any questions, I'd like to address those now.
Great, thanks Eloise. It's now open for questions. While you're thinking of your questions, I might start. So I'm intrigued with the presynaptic receptors, particularly on the glutamate input. And kappa receptors, dynorphin, the endogenous agonist, is co-expressed in the orexin cells. John Mushamp showed that many, many years ago. And there's a very, very significant orexin input into the PVT. So I'm just wondering whether or not, A, you know the source of those glutamate terminals, where they're coming from, and B, whether or not you've also looked at the cells that are sensitive to the kappa ligands, express a kappa receptor, also express an orexin receptor, because you'd expect that they would.
Yeah, thank you very much. That's an excellent question. There's two different questions here. So the glutamatergic input is coming from all over the brain, but yes, you're right, also from orexin neurons. Looking at orexin and kappa in the PVT, I've personally not done this, but Alessandro Mazzei published a beautiful paper in 2018 where they do show that there are also orexin and kappa receptors, and they counteract each other. They do exactly the opposite things.
Any questions?
I'm just going to jump in very quickly and ask you, because the question I was going to ask is, what prospect for kappa receptor antagonists in the treatment of drug-seeking behavior? And you obviously have a poster. I wonder if you could just give us a 1-minute preview of that in terms of what prospect for targeting kappa receptors in the context of substance use disorders.
Okay, yeah, thank you for that question. So my poster is actually looking at kappa agonists, and I showed that we can actually use kappa agonists to decrease drug-seeking behavior, which is counterintuitive, but it's only selective when you pair this very contingently to the originally drug-paired context, so counter-conditioning. I do think that kappa antagonists really show potential as they're like soaring through clinical trials, and I do think that especially decreasing the dysphoric and stress-related aspect of drug addiction using kappa antagonists is very promising, but I've personally not looked into this. I only look at kappa agonists.
Okay, well, if there's no further questions, thank you so much, Eloise. It was really interesting. Thank you. Yeah. Okay, and our next talk is from another good friend of mine, Dr. Markus Heilig, who is in Linköping. Oh, so my phone just went dead, sorry. Yeah, so Markus is the Vice-Chancellor's Professor of Psychiatry and the Director of the Centre for Social and Effective Neuroscience at Linköping. He has something in common with Paul in that he did his postdoc at Scripps in George Koob's lab. Paul wasn't in George's lab, but he was in a next-door lab. Since then, Markus has gone on to have some very senior positions, such as leadership positions at the Karolinska Institute in Stockholm, and also Director of Intramural Clinical and Translational Research at NIAAA in the States. And he left there to go back to Sweden to set up his team back in Sweden. He's won a number of awards, such as the European Neuropsychopharmacology Award, the Swedish Soderberg Award, and he's also an elected member of the Swedish Royal Academy of Sciences and a Wallenberg Clinical Scholar. And we're going to hear now Markus's research into GABA-B receptors for the treatment of alcohol use disorders. Thanks, Markus.
Thank you very much, Andy. So I guess after this kind of introduction, it can only be downhill. So thanks for that. I've also conveniently lost my voice, so we'll see how this will turn out. This will be a little bit old school. So GABA-B receptors have been around as a target for a while, right? But here's a new spin on it. So you saw my official title that's up on the screen. This is my unofficial title. It's a quote from the famous neurophilosopher Brian, AD 32. And you know, those who have seen the Monty Python movie know what it's all about, right? And this is to emphasize the importance of individual differences in studying addiction mechanisms in search of targets for new therapeutics. And I'll illustrate that with two of the strategies that we've applied in that search. So I hope you'll see that connection. You know, you heard some of this in Walker Lee's talk already, but I have to re-emphasize it. There's a lot of attention paid to the opioid crisis in the US, and that is, of course, for a good reason. Having said that, once you aggregate the morbidity and mortality from alcohol across the many users of that substance, actually the impact dwarfs that from illicit drugs combined. So we really have good reason to do better in search of therapeutics. And we're not doing all that well. This again is a reiteration, but this is my version of it. So, you know, we keep publishing all these circuit-busting papers in fancier and shinier journals. So that's that. I stopped counting the exponential growth of the neuroscience of alcohol addiction around this time. But we're developing fewer and fewer, or getting fewer and fewer drugs approved for that indication. So that's a disconnect we have to do something about. And this is a very sad graph because this is essentially Rajita Singha's replication of a study almost 50 years prior by Hunt and colleagues in '71, which showed that over about 1 year after detox, somewhere around 60 to 70% of alcohol-addicted patients relapse, and Rajita replicated this by studying her population, and the numbers have barely budged, right? So we need to do better, and it's kind of humbling to see that meanwhile, while we're doing all this fancy neuroscience, in an internal medicine department of places, where a young Lorenzo Leggio, under leadership of Giovanni Addolorato, are desperately trying to figure out how to help patients awaiting liver transplants to survive and do so by reducing their alcohol use and getting them in shape for transplants. And they try old, good baclofen without really having any clue about how it might be working. Giovanni had done some studies to show that baclofen could reduce withdrawal intensity in alcohol-dependent patients, and based on that, they just tried it. It's not a huge study, but you can see the benefits are pretty impressive. This is proportion non-relapsed patients. So this made the Lancet, and no clue about mechanisms, and also a fair amount of controversy because there have since been studies back and forth, some saying, yes, it works, others saying, no, it doesn't. So I think by now the dust has settled. This is an authoritative meta-analysis by Wim van den Brink and colleagues already 5 years ago, but there's been additional evidence since from J.C. Garbett just a year and a half ago published. So what you see here is the analysis of the meta-analysis of the studies by the Dutch group, but what that shows, in addition to supporting the overall efficacy of baclofen in alcohol addiction, it also shows you a very interesting relationship where the severity of dependence is an important moderator of that efficacy. So basically, if you start out with patients with high severity, you have a high effect size, and at the low end, and this is a non-replication by JC early on, and that was in patients with much lower severity. And JC then graciously, rather than debating the way academics normally would, I'm right, Leggio is wrong, JC went back to the drawing board, redid his study, but now selecting for patients with a higher severity of addiction, and lo and behold, replicated the Addolorato and Leggio results. So I think, it works in patients with high severity of addiction, but the question is, how does it work? And, how can we capitalize on this? Because I can tell you right at this point, baclofen is not going to be the therapeutic that we want. So in order to pursue this, I would like to say we did all this stuff. The reality is we did all this stuff, and then we realized that this is how it relates to the things I just told you. But, you can tell the story different ways. And so, first and foremost, we need to deconstruct the complex clinical phenotype of being alcohol addicted. It's just too broad of a category for me to understand, at least. And so we've tried to extract some key phenotypes, behavioral traits that we could hope to define well and then bring back into our mechanistic animal models. One of those is the progressive shift of choosing drug over non-drug rewards. This is, of course, Serge Ahmed's contribution in the basic science. He brought it back from actually Sprague and others who did studies in the 40s showing that under the conditions of availability of alternative reinforcers, you see very different behavioral phenomena than when you offer the drug as the only reinforcer available to the animal. So this is also telling the story a little bit backwards because we did this first in rats and then in humans, but this is the humans first. So you see heavy drinkers, and they basically classified according to the World Health Organization questionnaire that we use clinically. So this is the alcohol use disorder identification test. And we've designed pretty much emulating Lee Hogarth's work, but making it MR compatible so we can study the brain mechanism. Irene Peperini in our lab improved on Lee's model. This is what people do. We parametrically vary the point cost of working for one reinforcer versus the other. What you can see here, there's a ton of data from here, but one of the most important and striking results is the very strong correlation between the severity of alcohol use disorder problems in the wild and the frequency with which people choose alcohol over the non-drug natural reward in this model. What you can also extract from this is demand curves, or the equivalent of demand curves. And this is where it gets interesting in ways that I don't have time to go into, because as you probably know, and as we've heard a little bit of today, there's a lot of focus in the addiction field on habit formation and how supposedly once you develop these habits, you become insensitive to cost. And that is not what our findings are, and Lee's findings are very similar. We see that both these groups, at least, are sensitive to in this case, this is opportunity cost, right? Because they can get points for one reinforcer or the other, but this really can be transformed into demand curves. And you can see that while there is this shift in, if you will, affinity or intensity of motivation, there is still a sensitivity to cost. Now, the slopes are significantly different, so there is a different price elasticity, and that's the topic of the next paper that I'm not talking about, but this is not habitual. This is a shift. All right. So trying to bring this choice behavior into animal models then, in order to identify and pursue molecular mechanisms. This is published stuff, so I just summarize it in one or two slides. So some 5 years ago, Eric Kuijer, who came to us from Serge's lab as a postdoc, implemented this discrete choice model. He trained rats on regular self-administration first, so under single reinforcer conditions. Once everybody has acquired self-administration and they self-administer at pretty much same rates, you now put them into this discrete choice model. They first have to figure out that one lever will deliver alcohol, the other highly palatable saccharin solution. And once they figured it out, you now introduce this mutually exclusive discrete choice situation where they can press either lever, but as they do so, and receive that reinforcer, the other lever is retracted, so now they have foregone that reinforcer in favor of the one they chose. And you keep cycling this over and over again. Here's where you see the importance of individual differences, because if we only calculated group averages, this would be a very short story, and the story would sound, rats don't particularly like alcohol once given an opportunity to choose something else. And that's totally in line with Serge's conclusions for cocaine and for heroin, right? Shows rats lever press hundreds of times for cocaine. You'd think they'd continue to do so no matter what else happens in their lives. It's enough to offer them a second lever that delivers a sucrose or saccharin solution. They stop, and as a group average, will switch over to pursuing the natural reward. But if you disaggregate this into distributions, you see a very nice bimodal distribution where, yes, the vast majority of the rats will switch over to the natural reward, but there is a substantial minority that, as a stable individual trait, show a choice preference for alcohol. They're about 15%. So it kind of doesn't make sense to look for mechanisms of alcohol addiction in the total population of rats, which on average does not show a choice preference for alcohol. We think we might be more likely to identify those mechanisms by focusing in on the minority that actually does choose alcohol. So that's what we did, and a long effort using differential gene expression analysis across a bunch of brain sites that I won't bore you with now. Interesting result. My favorite structure that I've been trying to get away from for about 25 years now since I did my PhD, the central nucleus of the amygdala was the only one that popped up with a significant number of differentially expressed transcripts, and among those we found a whole pathway of GABAergic genes, and it all seemed to reflect a downregulation of this GABA transporter, which here by Rainer Spanagel in his commentary was drawn as being located presynaptically in a neuron, and that is correct for about half of them. What Rainer missed out is what should have been here is the astrocyte that expresses the other half, right? So actually, we have no clue whether it's the astrocytic or the neuronal GAT3 that matters, but what does matter is that this results in this hyper-GABAergic state in the central nucleus, and it's very clear that if you're able to normalize that, you can rescue choice behavior, and conversely, if you take rats that don't choose alcohol and you knock down the GAT3, you will shift them towards being choosers, right? So here's indication that GABAergic transmission in the central nucleus important for one key component phenotype of the broader phenotype of alcohol addiction. And then the other one we think is important, and as a clinician I'm becoming more and more convinced the longer I work with patients that this is at the core of the disease, is continued use despite knowledge of adverse consequences. This is one of the DSM criteria, but there it's one of 11. I think this is really fundamental. People know that once they have a liver cirrhosis, if they keep drinking, they'll die. They keep drinking and they die. Okay, that's continued use despite knowledge of negative consequences. So again, very similar approach, reverse translation. So Erika Grodin, when she was brilliant graduate student at the NIH, now with Lara Ray at UCLA, Erika did this heroic effort for her thesis where, again, she brings in heavy and light drinkers. So all the heavies are up here, all the light are here. You may not see the distinction, but what you see here is another rating scale for compulsive drinking score. It actually correlates very strongly with the audit that we happen to have this one. It's pretty much the same. How much problems do you have with alcohol in the wild? And that correlates very strongly with continuing to button press for alcohol points under conditions when you are signaled that, yeah, you can do this, but you are at a 75% risk of receiving an electric shock. Yeah, Erika did shock people. It's not as bad as it sounds. They get to titrate out the shock intensity themselves. But it's remarkably similar to what we see in rats, right? So a bunch of people will suppress the behavior. That's the light drinkers. A bunch of people will not. That's the heavy drinkers. We have a bunch of brain imaging data that I actually will just cut out from that study. But the fundamental observation is, yeah, you can model the phenomenon of continued use despite knowledge of negative consequences in the lab in humans. And so then we take it back into the rat, again, as a reverse translation in order to do the things we can't do in awake, freely moving humans, which is go mechanistic. And so, very similar strategy, again, focus on individual differences. Remember the neurophilosopher Brian, right? We're all individuals, and so are the rats. And so, in this case, Essie Domi, now back as faculty in Camerino, but a wonderful postdoc in the lab for 5 years. Heroic effort, she, again, trains large populations of genetically heterogeneous Wistar rats on self-administration the way we always used to do it. But once that's done, she introduces the negative consequences as a 0.2 milliamp foot shock. And actually, initially, not a whole lot happens. You see a spread, but it's just unimodal normal distribution, so that seems pretty random. And I think that's one of the reasons why a bunch of labs have tried this and given up on it. But you need to have Essie's persistence, and if you do, and if you keep going for 2 weeks, you see this beautiful segregation. It's shown even better here. You see this clustering into two populations where 2/3 of the rats will do what you would expect a sensible rat or human to do once negative consequences hit, i.e. suppress the behavior, right? So that's these guys. But then you have this about 1/3, or thereabout, that just couldn't care less. And about 15 different control experiments, of course, to exclude that this is due to differential sensitivity to the shock-induced pain and other confounds. But this is actually real. This is correlated with choice behavior. We saw this already in the choosing rats in the science paper. And it also, as we show in this paper, correlates very nicely with the other model of compulsive or aversion insensitive alcohol, pursuit of alcohol that is widely used in the field, which is drinking quinine adulterated alcohol solution. Except this one is much more sensitive and parametric, so we prefer this one. But they correlate 0.8. So having identified this subpopulation, again, we embark on this 2-year discovery effort. In this case, we do so by whole brain FOSS mapping in order to identify neuronal populations that are active as animals compulsively self-administer alcohol. So now you have to parse this out because obviously we're not interested in neuronal activity that's associated with just receiving shock. So now you have to have a yoked control group, and it's a lot of work. And as you'll see in a moment, we could have saved ourselves all this trouble have I not so hard wanted to get away from my good old PhD thesis structure and had Essie Domi not so much wanted to find a structure of her own rather than pursuing what Eric had already found. Because what happens, once I could show you bar graphs up and down for individual structures, but what we like to do, in my other life, when I do brain imaging, we're interested in networks. We do this as a network analysis. It's simpler because obviously there's no time dimension. So just run a principal component analysis, we identified two networks of intrinsically correlated neuronal activity that's associated with compulsive alcohol self-administration. But the remarkable thing is that one of these two networks, while closely correlated with the compulsive self-administration, does not discriminate between the compulsive and non-compulsive rats, while this one very much so does. And guess what's at the hub of this network? It's called the central nucleus of the amygdala. And like I said, we could have saved ourselves a lot of trouble because actually we've done more rats, and this correlation is R squared, 0.75. I've never seen a correlation between neuronal and behavioral data like that over the course of my entire career, right? So 75% of variance in compulsive self-administration accounted for by neuronal activity in the central nucleus of the amygdala. I don't know how much you can see in this light, but these are the neurons. I can't see much from where I stand, so sorry.
I can see them, it's okay.
Okay, good. So I'm not making this up. I'm past doing correlation neuroscience, though. We got to get causal. So it's nice to see that these ensembles are activated while this is ongoing and seeing the strong correlation. But asking the question whether it is actually activity of these ensembles that is causing the behavior, it's a much harder question to ask. But by now we do have the tools to ask those questions. We're a little bit worse off doing this in rats than our friends studying the lesser rodent, because we don't have any driver lines. But with a two-vector system that was provided to us by colleagues from Amsterdam, Michiel van der Hoven, you can tag all the neurons that are active while the animals engage in compulsive self-administration, and you can get them to selectively express an inhibitory dread. So that's what we do, right? So I won't spend time on the procedural details. We can discuss those in a Q&A if people are interested, but this is basically a modified Cre that is driven by a FOS promoter in one of the vectors, and then a Cre-dependent inhibitory dread in the other one. And the trick is titrate the viral titers so that you get a balanced expression of the two. And we got lucky. I can tell you my big brother Yavin Shaham tried this same system for a year and a half and told me not to give it a shot because it was never going to work. So we were surprised. It did work. We could express the dread in the ensembles. The question, of course, is now if you administer the actuator of the dread, the testable prediction is if the ensemble activity is driving the behavior, then activating the inhibitory dread should markedly suppress the behavior, and that is in fact what you see. This is the only thing you need to look at. So these are the rats that express these are the rats that express the inhibitory dread, and they drop down by over 50% in their compulsive self-administration, and the guys that carry the empty control vector are completely unaffected by the CNO, which nicely controls for the possibility of non-specific effects of CNO, which we now know might mess these things up. Okay, so yeah, the activity of these ensembles is causal, and so who are these guys? It's nice to have an ensemble, it's nice to show causality, but if you want to do pharmacology, we need to understand what is the cell population. And so this all happens in the centrolateral division of the central nucleus, and 80% of the cells in that structure they're all GABAergic. They're not all GABAergic. Most of them are GABAergic. Among the GABAergic neurons, about 80% are made up by two populations, one that expresses the neuropeptide somatostatin, the other one expresses the signaling enzyme PKC delta. And what you see is that all the ensemble members are in the PKC delta positive neurons, okay? And why is this interesting? That is because within the centrolateral division, you have this yin and yang organization, or a seesaw organization between the PKC delta and the somatostatin populations, and basically the balance between two, according to at least one conceptualization, which has gotten a little bit more complicated since, but I still think this has some important meaning. The balance between these two gates the outputs from the central nucleus, which originate in the central medial division and then run to a bunch of structures, one of which is the PAG, which controls inhibition of behavior, right? And of course, what you need to do if you are pursuing a reward and now are provided with information that the pursuit of this reward will get you in trouble, you need to put a brake on the approach or drug-seeking behavior. And so this could be one such brake. And if that brake is lifted, that is when you would see aversion-resistant pursuit of alcohol. That's my working hypothesis at the point. More importantly, it turns out, I may have lost the slide here. So all the PKC delta neurons express GABA-B receptors. Pretty important slide. Sorry I lost it, but now you have to take my word for it. And what happens if you activate those GABA-B receptors is that you will, through microinjections of baclofen locally in the central lateral division of the central nucleus, you will inhibit both the activity of neurons as seen as colocalized FOS and PKC delta expression, right? This is a pretty drastic inhibition, and that is accompanied, as would be predicted, by a decrease in compulsive or aversion-resistant alcohol self-administration. So, GABA-B receptors are pretty abundant, right? And the obvious question is, yeah, it's nice that this works in the circuit in the central nucleus. What about the 2 million other places where GABA-B receptors are located, and what is going to happen if you administer something that activates GABA-B receptors at a level that will attenuate compulsive self-administration? What will happen to all the other functions that may be controlled by GABA-B receptors? And luckily, there is a window where you can significantly and robustly suppress, even with systemic administration of baclofen in this case, you can suppress compulsive self-administration without really messing up locomotor activity, self-administration of non-drug reinforcers, etc. It's very clear that once you go up in dose, you will get sedative effects, and this gets me to why baclofen is never going to be the medication we really want, right? So there's a bunch of boring reasons why that's not going to happen. It's off patent, no one can make any money off it. People have tried reformulating it, extracting the active isomer. That's not the major obstacle. The major obstacle is baclofen is an orthosteric agonist. We know what to expect when we kick a GPCR with an orthosteric agonist day in and day out. It will induce tolerance. And indeed, that is what we see. We can see a reasonably nice and potentially useful therapeutic window on acute treatment with baclofen, but it will require in many patients escalation of the dose, and that is indeed what happens. A typical daily dose that we use is 35 to 70 milligrams. And some patients do well with this. We do use it. A lot of patients will escalate to a couple hundred milligrams a day. These are doses where we know that sedation, other adverse events increase in frequency. There have been deaths when people have escalated to those levels. And based on that, the European Medicine Agency has turned down baclofen on the indication for alcohol addiction. Now, there is an exemption in France, I believe, but that's France. I think we can, now that we understand how activating GABA-B receptors really target some of the core mechanisms of alcohol addiction, i.e. the choice preference for alcohol over natural rewards, and the compulsive self-administration that continues despite knowledge of negative consequences, it becomes a pretty important question. Can we somehow capitalize on this mechanism without getting in all the kinds of trouble that baclofen gets us into? And a potential answer to this question is GABA-B positive allosteric modulators. So quite some time ago, we published, again, a skipped slide, but we were approached by a small Swiss medicinal chemistry company, ADEX, and asked whether we'd like to evaluate their GABA-B PAM at that time, 10 years ago, 12 years ago, I forget. We had no clue about any of the stuff that I'm talking about today. We evaluated it in a bunch of other assays where it actually works quite nicely as well. as it happened, once we were doing the choice work and all that followed, we realized, oh, wait a second, we have some leftover ADX71441 in the drug cabinet. We pulled it out, was literally the last milligrams we had, and so we ran the experiment twice for technical reasons, and you can see how beautifully the GABA-B PAM rescues the alcohol choice in the minority of rats, about 15% or so, that on the initial screen show a choice preference for alcohol as a stable individual trait without really doing anything to the majority rats that don't choose alcohol. So that was all great. We thought, this is the low-hanging fruit, right? We have fancy favorite targets that we would like to see develop, novel mechanisms, but this supposedly is low-hanging fruit. There's already one medication out there. How hard can it be to improve on it? Yeah, hard. So based on our data, Indivior licensed in ADEX's program, went into clinical development, molecule died in tox, right? But the tox was off target. There is now a program at Indivior and with ADEX to develop novel molecules. We are partnering with them, and Eric Augier, who has since gone on to become faculty in our center and runs his own group, is the custodian of that collaboration. So again, yet another piece of data that cannot be disclosed, but yes, there is now a selected clinical candidate after screening a bunch of molecules that looks every bit as good as the one I showed you, in fact, a lot better, and hopefully will make it through tox. Last minute, we need a biomarker. Meanwhile, I'm thinking, how the heck are we going to accelerate the clinical development? And this brings us to the need for biomarkers. If you have a PET ligand, you're good, right? But how else will you otherwise advance a molecule like this without all the expensive and time-consuming dose-ranging studies that people are not necessarily ready to invest in when it comes to an indication like alcohol addiction. Also, we've tried to be clever about this, and it turns out if you use TMS combined with either EEG or EMG, you can isolate GABA-B function through what is essentially a paired pulse inhibition procedure. You buzz people first with a conditioning pulse, and then you come back with a probe pulse. And this decrease between the two pulses turns out to be mediated by GABA-B receptors. And in fact, in collaboration with Chinese colleagues in Shanghai, we found that GABA-B mediated inhibition is impaired in people with alcohol addiction. So, that's a story for another day. That's published in Molecular Psychiatry. So, we think that we have we have a mechanism, we have molecules, we have the biomarkers. Let's just hope that the tox studies play along. With that, I'll sum up. So, one important lesson I've learned is that the animal models that I at least have used for ages to try to discover new targets and therapeutics, not necessarily all that useful. Most rats will not choose drug over a natural reward. Most rats will not continue to self-administer when punished. But there is a stable minority of rats that will show these behaviors, and that really looks to us like an addiction-like syndrome of traits. I didn't have time to show you the other behaviors, but they have increased motivation, they're more prone to relapse, and so on. And using that population, we repeatedly stumble upon GABAergic mechanisms in the central nucleus of the amygdala as a potential target, and it looks like GABA-B activation can rescue it, both alcohol choice and compulsivity. GABA-B PAMs may get us around the limitations of baclofen, and TMS-based biomarkers have the potential to accelerate clinical development. So with that, I stop and thank all the people who do the work and take questions.
Thanks, Markus. Very interesting. We've got time for a couple of questions. I might start with one while someone's heading to the microphone. Have you thought of looking at a spatial biology approach to answer the GAT3 question in the neurons versus the astrocytes in the central amygdala in your alcohol choice rats, and then tagging on to that, have you looked at GAT3 expression in the ensemble cells in your punishment-resistant rats?
We considered whether to do, 3D sequencing, or so we're right now actually running the single nucleus RNA sequencing effort from compulsive rats to address exactly those questions.
Yeah. Okay. Very interesting.
I have a quick question, if I might. We know that not all positive allosteric modulators are created equally. For some receptors, you only need to shift agonist concentration response curve twofold. For some, you need to shift it 100-fold. Can you give us any insights into what is the mechanistic pharmacology of the ADX compound that you presented?
I'm embarrassed to say that I can't. I don't know. Sorry.
That's okay.
That's right. Okay, last quick question, Dan.
So, Markus, have you tested any psychedelic like LSD or DOI in your model? I mean, the crazy rats?
So I have half the lab bugging me to stick psilocybin into our compulsive rats, and so far I've resisted it, but I may not be able to do so much longer.
What's wrong with you?
All right, so thanks very much, Markus.
Thank you.
Okay, thank you, Markus. Our final speaker of the session today is Kari Johnson. Kari is from the Uniformed Services University. She did a PhD at Vanderbilt and a postdoctoral training at NIAAA. And today she's going to talk to us about mGlu2 receptor modulation of reward and reinforcement driven by thalamostriatal circuit activation. Kari, over to you.
Thank you. I really appreciate the opportunity to speak here today. Before I start, my employer requires me to tell you that these views are my own and I have nothing to disclose. As a lot of the people you've heard from today, there's a big push in neuroscience these days to understand how specific types of cells and specific brain circuits contribute to behavior. And as somebody who's interested in both circuit neuroscience and pharmacology, I really like to think about ways that these two areas can intersect, especially in terms of understanding how the mechanism of action of drugs and how they affect behavior. I think this is important for a couple of reasons. I am a naturally curious person. I want to understand how our drugs are having their effects. But I also am very hopeful that having a more detailed understanding of how drugs act in a rodent brain in preclinical models and in a human brain could actually help us improve translational prospects, because as we all know, especially in the neuroscience field, a lot of drugs fail to translate from preclinical efficacy to efficacy in human disease. So I think today I'm actually going to tell you a story that takes a little bit of a different route, though, where we had a drug where we're interested in the mechanisms of action at a synaptic level, and it's actually led us to think about a brain circuit in the context of reward and reinforcement and addiction that really hasn't received a lot of attention in that area before. So I'm going to be talking to you today about the GPCR Metabotropic glutamate receptor 2, which is a long-time love of mine. And I am really interested in mGlu2. One reason is because it's a target for treatment of substance use disorders. So there's currently positive allosteric modulators that are in clinical development for treatment of substance use disorders, particularly first line for nicotine use disorder. And the reason for that is that there's a lot of preclinical evidence that both on an acute time scale and when thinking about chronic drug use, that mGlu2 can modulate drug-related behaviors. So on a more acute time scale, if you give an mGlu2 agonist prior to a psychostimulant, which we know evokes excessive dopamine transmission in striatal regions, mGlu2 can actually inhibit that drug-evoked dopamine release. And in line with that, you also see an inhibition of the psychomotor activation that comes with a lot of psychoactive drugs. In more complex models of chronic drug self-administration, when you give an mGlu2 agonist in both rodents and non-human primates, there's reduced self-administration of a whole variety of different drug classes, including alcohol, cocaine, nicotine, and amphetamines. And in addition, when animals have been allowed to go through a period of extinction or abstinence and then reinstate their drug self-administration, mGlu2 activation in some cases can reduce that reinstatement of drug seeking. I've shown an example from Athena Markus' lab on the right here of nicotine self-administration where mGlu2 PAM, AZD8529, dramatically reduces the self-administration of nicotine over a period of several weeks with no tolerance forming. And I should point out here that a lot of these studies were done with drugs that do not distinguish between mGlu2 and the closely related mGlu3 receptor. But more recently, there's been development of selective positive allosteric modulators as well as transgenic rat and mouse lines that have allowed mGlu2 to be identified as the receptor subtype that's really the most important when thinking about addiction, especially in the context of the striatum, which I'm going to talk to you about today. So what does mGlu2 do at a synaptic level? mGlu2 is usually expressed presynaptically. It's a GIO-coupled GPCR, so we think about it reducing cyclic AMP accumulation. But importantly, at the synapse, it can also modulate ion channel function. So here we have voltage-gated calcium channels, and mGlu2 can inhibit opening of these voltage-gated calcium channels, which reduces calcium influx into the presynaptic terminal and reduces neurotransmitter release. So if we're thinking about how mGlu2 might affect psychoactive drug-evoked dopamine release to start, it's interesting to think that, well, maybe the dopamine receptors express mGlu2, and that would be a straightforward way for them to reduce that drug-evoked dopamine release. However, there's no evidence that mGlu2 is expressed in dopamine neurons. And here's some functional evidence of that from Beta Mogadam's lab many years ago now, where they gave rats L-DOPA, the precursor to dopamine, and measured extracellular dopamine levels in the striatum. And you can see that L-DOPA administration causes this increase in dopamine transmission. But in this case, if you give an agonist of mGlu2 and mGlu3 before the L-DOPA, unlike with psychoactive drugs like stimulants, you don't see a decrease in dopamine transmission. So what this tells us is that it's likely that mGlu2 is actually acting through some sort of indirect mechanism to modulate dopamine transmission, and I've always been very curious about what that indirect mechanism might be. So to get at this, we started thinking about the different inputs to the striatum. So here I'm showing you the rodent terminology. This is a coronal slice of a rat striatum or a mouse striatum. We have the dorsomedial subregion, which is mostly associated with goal-directed behavior, the dorsolateral striatum, which is mostly associated with habitual behavior, and then the nucleus accumbens, which is famously known for its roles in reward processing and motivation. Although I should really point out here that these different functions are not that cleanly segregated by any means. Each of these regions receives glutamatergic inputs from a different variety of cortical regions that have functions associated with the functions of the striatum that they're projecting to. And also they receive major glutamatergic inputs from the thalamus, which relative to corticostriatal transmission has been largely ignored. And then of course, these areas are all innervated by midbrain dopamine neurons as well. So we started out by doing some wholesale patch clamp electrophysiology to start trying to figure out which particular synapses were being modulated by mGlu2. So we did patch clamp recordings first in the major projection neurons in the striatum, the medium spiny neurons, and used electrical stimulation. So here we're non-specifically activating these glutamatergic inputs to these neurons. And when we apply an agonist of mGlu2 and mGlu3 to our slices, it causes this robust long-term depression of excitatory transmission. We did a paired pulse ratio test, and the bottom line here is that when you see this increase in the paired pulse ratio, it suggests that there's a decrease at the presynaptic level of neurotransmitter release probability, kind of confirming that it is likely to be a presynaptic site of action. As I said, this is an mGlu2 and mGlu3 agonist. We were fortunate to obtain mGlu2 and mGlu3 selective negative allosteric modulators from Jeff Kahn and Craig Lindsley at Vanderbilt. And we were able to show that the mGlu2 NAM completely blocks the effect of the agonist to inhibit excitatory transmission, whereas the mGlu3 NAM is not effective there. So this effect is exclusively mediated by mGlu2. Now, I showed you a very non-specific way of activating synaptic transmission to study it here, but we were interested much more specifically in whether cortical inputs were affected by mGlu2 or thalamic inputs. So to look at this more specifically, we used an optogenetics approach. So here we're virally expressing an opsin called channelrhodopsin 2 in neurons that we are interested in activating. This makes them light sensitive, so then we can flash blue light on these striatal slices, and then we'll get glutamate release only from those neurons that are expressing the opsin. So we did this both in, this is just a couple of examples. We've done this in a lot of regions, but here I'm showing you in the primary motor cortex as well as the thalamus. And you can see that when we selectively activate either corticostriatal transmission or thalamostriatal transmission, in both cases we see still this robust long-term depression of excitatory transmission. So we didn't see any selectivity for mGlu2 modulation of either of these pathways, but we hadn't really thought about what it might be doing at these thalamostriatal synapses before. As I've said, they are relatively understudied compared to corticostriatal synapses. So we started to think back in the literature about what these thalamic projections to the striatum do. So the regions in the in the thalamus that primarily project to the striatum are these midline structures. Here I'm showing the more anterior ones, so the centromedial nucleus, the paracentral nucleus, and the centrolateral nucleus. So these provide glutamatergic inputs to the dorsal striatum that drive the activity of striatal projection neurons. And it's really important for me to point out because striatal neurons are very quiet on their own. They really need glutamatergic inputs from the cortex or the thalamus to drive their activity. So this really has the potential to have a major impact on striatal output. Now, something else that's really interesting about these thalamic inputs to the striatum is that they can drive a non-canonical form of dopamine release that I will explain more to you about in a moment. But that's something that our lab has been really interested in. And then decades ago, when people were using the ICSS type of paradigm that Paul was discussing earlier to kind of map different reward centers in the brain, it was discovered that if you put an electrode into these areas, that rats will self-administer the electrical stimulation of these areas, suggesting that activation of these areas in a fairly nonspecific way can be rewarding. So here's a schematic that will help me explain this non-canonical dopamine release I was referring to. So we're looking in the striatum here at these thalamic inputs. So they release glutamate, and they activate this population of interneurons in the striatum called cholinergic interneurons. This is a fairly small population, only 1 to 2% of striatal interneurons, or striatal neurons, but they are extensively ramified and have a very strong modulatory effect on various striatal physiology. So one of the interesting things that they do is when they release acetylcholine, that can act on nicotinic receptors that are near release sites on midbrain dopamine neuron terminals. And it triggers a local action potential that causes dopamine release that is completely independent of the somatic activity of these dopamine neurons. So we wanted to know if mGlu2 could also regulate this non-canonical form of dopamine release, and so first we did whole cell patch clamps and patch clamp in these cholinergic interneurons, and we saw that activation of mGlu2 could again, cause a very robust depression of synaptic transmission. And then when we used an electrochemical technique called fast-scan cyclic voltammetry to look at dopamine release, this is being triggered by optogenetic activation of these thalamic inputs, then we see that compared to baseline, activation of mGlu2 causes a really strong reduction in that dopamine release. So this finally gave us at least one mechanism that mGlu2 can modulate striatal dopamine release. So then we wanted to ask what this means on a behavioral level, and we really had to take a step back to think about what mGlu2 might be doing at in this pathway, because there really hasn't been a lot described about what these thalamic inputs to the dorsal striatum do. And so the first question that we asked, because we know that dopamine release in the striatum can be reinforcing, and we know that activation of the projected neurons can be reinforcing, we thought it stood to reason that thalamic inputs to the dorsal striatum could also support reinforcement. And so we set up an optogenetic version of the intracranial self-stimulation model to test this. So we used a viral injection to express channel rhodopsin in the thalamus, and then implanted optical fibers in the dorsal medial striatum. And then mice were allowed to press a lever on a FR1 schedule, so one lever press for one stimulation train, to receive this stimulation. And we were simply asking the question, will they press the lever for this stimulation? So in what I'm showing you, they're receiving a 10 hertz, 10 pulses stimulation, so 1 second at 10 hertz. And you can see that this elicits excitatory postsynaptic currents in the projection neurons in the striatum, as well as a very robust dopamine release that is completely sensitive to the nicotinic receptor antagonist DHβE. So as you can see on the left here, the mice that were expressing channel rhodopsin in the thalamus escalated pressing on the active lever for this optogenetic stimulation of the thalamostriatal terminals, but did not press the inactive lever. And then in our control animals that were just expressing a fluorophore, they did not acquire lever pressing. So it does in fact seem in this paradigm that thalamostriatal stimulation is reinforcing. I'm showing you here individual lever pressing patterns from 3 different mice that were in this study, just to show you that they kind of press in a pattern. So they tend to have a cluster of presses and then take a break, and I'll come back to that in a little bit. So of course we wanted to know what mGlu2 activation would do this, and we kind of predicted that mGlu2 would reduce the reinforcing value of this stimulation because it reduces both glutamatergic drive to the projection neurons and also dopamine release. So here we pre-treated the animals before their session with the mGlu2-3 agonist, LY379268, and you can see that there was a very robust reduction in lever pressing here. We also did the converse experiment and pre-treated the animals with an antagonist to see if there would be any effect. And we actually saw this really nice increase in lever pressing. And so this suggests to us that mGlu2 is being tonically engaged when the animals are performing this task and receiving this stimulation from thalamostriatal transmission. And that is tonically constraining the reinforcing value of this stimulation. We also compared this to responding for a natural reinforcer. This was in a separate group of mice. So here, when we gave the antagonist, we were actually surprised to see about a 50% reduction in responding for food, which was the complete opposite of what we saw with the optogenetic stimulation. And then we saw a very weak inhibition of responding for food with our highest dose of LY379268, about 10%. It's a modest effect, and we've done this with other reinforcement schedules like random interval schedule and actually not seeing any effect of this dose. So it seems that the effect of mGlu2 modulating lever pressing here is very specific to the reinforcer. Now, I mentioned that the animals press the lever in this distinct pattern where there's clusters of pressing separated by breaks. We don't really know what this means, but we wanted to know if there was a specific way that mGlu2 modulation was changing these patterns of lever pressing. So we did an analysis looking at the number of press clusters per session, the numbers of presses per cluster, and then the duration of each cluster and press rate within each cluster. So what we saw was that LY379268, the agonist, decreased the number of clusters per session, but when we looked at these within cluster parameters, the duration, the number of presses, or the press rate, we didn't see any effect. So it was changing the animal's approach to the lever, but not changing the way that they were engaging with the lever once they did. We saw the exact converse of that with the antagonist, LY341495, where it increased the number of clusters per session, but not these within cluster press rates. So we're still not entirely sure what this means, but it does seem that there's a very specific way that mGlu2 is adjusting how the animals respond to this stimulation. So we also wanted to ask if the stimulation is rewarding. Now, a lot of times people will use the term reinforcement and reward interchangeably, but animals do sometimes press a lever for something that isn't rewarding, so we asked this kind of more specifically using what's called a real-time place preference task. In this task, the animals are allowed to freely explore a chamber. There's two chambers that are decorated with different visual cues on each side, and they're separated by a small hallway. So this is a 5-day paradigm where the animals on the baseline day are allowed to freely explore the chamber. And then on all days, they're allowed to freely explore the chamber, but on days 2, 3, and 4, we give optogenetic stimulation of those thalamostriatal terminals in the dorsomedial stratum when they're on the side that was originally not preferred to see if they'll seek out that stimulation. And then on the final day of testing, we stop the stimulation and see if they've developed a conditioned place preference based on the stimulation. So what we saw here is in the circles, you can see that on the days that the animals received stimulation, they strongly preferred the stimulation paired side, which was the originally non-preferred side. These were the mice expressing chenorodopsin, and the control mice expressing the fluorophore. YFP did not develop this preference for the light stimulation. So this does seem to be a specific effect. Now, the real-time place preference lasted for all 3 days of stimulation, but as soon as we discontinued that stimulation, the animals no longer preferred the stimulation paired side. So we did not see a conditioned place preference under these conditions. And then we wanted to know what mGlu2 would do to this place preference as well. So we used a within subject design here. Since the animals didn't maintain the preference, that allowed us to repeatedly test the same animals for their seeking of the stimulation. And so we had animals in a counterbalanced order given either vehicle or LY379268 prior to the session. And you can see here that the animals preferred the stimulation paired side much less on their LY379268 day compared to their vehicle day. This looks like there might even be an aversion that develops to the stimulation when mGlu2 is activated. But this is something that we'd have to follow up on with a more specific test for that. So what I've tried to talk about today, which I know is much more basic science than a lot of my colleagues in this symposium, is that we've used this pharmacological finding to kind of think about what that might mean in the context of reward and reinforcement and addiction for thalamostriatal pathways, which really hadn't been explored much in this context before. We found that thalamostriatal activation can, in fact, drive behavioral reinforcement and reward, and that mGlu2 activation constrains that reward and reinforcement. And we're interested going forward in exploring whether this could be a possible mechanism of action in terms of the models of what's going on with psychoactive drug use. just on taking a step back from that a little bit, I think it will be really important to learn more about how psychoactive drugs affect the reinforcing and rewarding properties of thalamostriatal activation. There's likely to be some significant differences in the types of behavior that are influenced by dopamine that's coming from different pathways, and this is an area that we're just starting to scratch the surface on right now. So I'd like to thank members of my lab who worked on this, particularly Mike Hochstein and Doris Chang. I began this work when I was a postdoc in Dave Leventer's lab at the U.S. National Institute on Alcohol Abuse and Alcoholism. And I'd like to thank my funding from the NIH, and I'd be happy to take any questions.
Thank you very much, Kari. Whilst give people a chance to think about any questions that they might have, I have one. mGlu2/3 agonists have obviously been tested in clinical development reasonably extensively, not in the context of addiction, but are there any learnings or insights or even anecdotal evidence that have emerged from that clinical work that might support a role clinically?
In addiction, I don't think I've ever heard anything along those lines, although, I'd be surprised if there wasn't some sort of data buried somewhere on that, because it's the study populations have been, looking at schizophrenic subjects, and alcohol or substance use disorders are highly comorbid with schizophrenia. So it's likely that people would have been using substances, and that there might be some data there. There was actually a phase 1 trial with an mGlu2 PAM for smoking cessation. I don't know if the data from it was ever actually released. I don't know that I've ever seen the actual data. I've heard that it didn't meet its endpoint for smoking cessation, but was well tolerated in general. But there was a theory that there were some dose-limiting side effects so that the hypothesis maybe really wasn't tested. And so I don't think that there's much clinical evidence yet, but I know there's at least some groups who are working towards testing that more thoroughly now. And I think it might depend on which substance you're talking about, the efficacy, right?
There's no one else. So I'm going to go on that point. mGlu2 is just a classic autoreceptor. So you activate the autoreceptor, you dial down the glutamate release, and thereby ultimately get the endpoint that you record. But it's not only an autoreceptor on those thalamostriatal terminals. So in terms of, from a clinical development perspective, you're going to be activating autoreceptors on lots of glutamate terminals and toning down glutamate release in all sorts of places. And do you think that maybe is one of the sticking points in the clinical development of ligands of that nature?
I certainly think it could be. That's another reason more specifically that I think it's important to understand the mechanisms of action, because let's say we see that dampening the activity of one pathway is going to be efficacious, but when we more broadly dampen activity, it kind of cancels out the beneficial effect. Maybe we can find other receptors that do something similar in that same pathway, or maybe that are expressed in that pathway in humans, if the receptor expression is not the same between rodents and humans, and find another strategy that mimics it that might be more effective.
Shane.
Thanks for a really interesting talk. So mGlus are fun in that they can heterodimerize both with other mGlus and can form complexes with other classes of receptors as functional crosstalk, things like that. Yes. So because these synapses are relatively understudied, do you have any idea about the other complements of receptors that are there, and potentially how that would affect the pharmacology that you'd be looking for to manipulate the synapses?
Yeah, so there's certainly evidence that mGlu4 and mGlu2 can heterodimerize, and I think that based on some work that I was actually peripherally involved in back when I was at Vanderbilt when we were first looking into that, that at corticostriatal synapses it's likely that there is an effect of mGlu2 for heterodimers there. For the thalamostriatal synapse, like, no idea. we really haven't had a chance to look into that at this point. I think that mGlu4 is maybe expressed there, but we really haven't like functionally explored that yet. And it's tricky too, if, you know, until we have like better pharmacological tools that can maybe like very specifically target those heterodimers. It can be pretty challenging to ask those questions in native systems.
Okay, well, if there are no further questions, just before I ask you to join me in thanking Kari again, I would just like to thank all of the speakers for bringing together what I think we'll all agree has been a really fascinating and interesting symposium. So thanks to my co-chair, Andy, and all of the speakers, and Kari particularly. Thank you.